Yeah, I changed it to non-identical this morning.
Thank you.
3 magical letters and a hyphen, you know.
Yeah, because I made 92 the first time I'd taken it. I said, I know that 49 had to be right. Second time I take it, I got it right. I said, okay, maybe I can read through the lines.
Is there any way we can get copies of these to study from?
A lot of them are in the lecture, right?
Yeah, I know. I've had a lot of questions on there.
Mm-hmm. All right, it's just over the new material.
Yeah, I've been watching your fall 2003 lectures. I like them. Because, you know, I sit in the class and I want to rewatch those. So I watch those. They're a little different.
They are different, yeah.
Yeah. I think they're at Brooks or something.
Yeah, and the technology was a bit different.
Yeah, I kind of like them. They're a little different twist to it, I guess you'd say.
Yeah.
And you get a little bit more out of it because you can explain it one way, but you may not quite explain it exactly the same way.
That's what I find really interesting about having the 2 semesters recordings is one semester your frame of reference is going one way, and then the next semester it's kind of another.
I really liked it because what I did during spring break was I watched them all before I take the exam, and it really, really helped.
I try to watch them before I come in to do the lecture scheduled for the day to try to match it, but it never seems to happen, you know.
It's close, but not what I'm saying.
Well, this morning is a day, if you've walked across campus and saw the group out there erecting this gigantic multi-banner display about abortion. I don't know what group it is, but certainly it's Right to Life group. And for the record, in the future, this is March 21st, 2005. This reminds us that in this class, we study fact and established, strongly supported hypotheses, and we study terminology, such as using terms conceptus and embryo and zygote and fetus and neonate for all the appropriate terminology and reference to developmental stages. This course, obviously, thereby is not related at all in content and intention to beliefs and opinions. We leave those for individuals. We don't even think cover the topic of abortion in the class. And this isn't a purposeful evasion of a relevant social issue. It's more so conservation of time so that we can cover all the terminology, facts, and developmental stages and try to learn as much as we can. So I just thought I'd pop that reminder in there as I walked by and saw all the pictures of the different developmental stages and certainly some very disturbing images of aborted late embryos and fetuses. And so these are of great concern to all humans, of course, and we, by taking this course and studying this kind of material, are in a better position to understand some of the basic biological descriptors that can be used in relevant philosophical arguments about our moral and ethical beliefs related to the termination of living cells, forms of life, organisms in general. So I'm going to switch to the computer now, and we'll begin with lecture 21. Today, there's a bit of technical difficulty with the picture-in-the-picture, the PIP, so this lecture will not have the PIP. If I'm going to be waving my hands or something, I'll try to remember to come back to teacher camera to be full screen to make some illustrations. But you may have heard in the earlier discussion and comments that we were having in the class here at the 1604 campus, this complete lecture is recorded under the fall 2003 heading, and you can watch it there. And no 2 lectures, even though they're on the exact same PowerPoint slide presentation, seem to be the same, regardless of whether I watch myself and try to repeat it or not. So there's a big plus to having both of those available. Today, then, we're going to discuss the digestive system, basically with the focus on the foregut and the mesenteries. We've discussed the mesenteries We remind ourselves of the folding, the head folding, tail folding, and lateral folding. And they're changing things on me here. It just looked like I was doing some magical stuff. And we know that all of these foldings had tremendous effect on the morphogenesis of the embryo and impact development toward a completely formed fetus with all of the body cavities closed and the organ systems developing with reference or juxtaposition to many of these body cavities and changing the shape of the body cavity, closing them off, separating them, and so on and so forth. So that will be reflected in the images that we see here in this presentation today. And we recall that with the two lateral folds coming around, we use the metaphor, I guess, or weak analogy of the butterfly wings folding together in front of the butterfly and fusing together at the margins of the wings and closing off the body cavities. And our focus today, after those events, and of course a tremendous series of events taking place for the head fold and the branchial arches, formation of the face and the oral cavity that we've studied, our focus then is on the foregut, and we remind ourselves that the two balloons coming together to form the enclosed pericardial peritoneal canal system forms the dorsal and the ventral mesenteries, and that will come through today. So we're going to start first by looking at the normal adult appearance of the foregut structures, and then proceed to a brief discussion of the events that occur in the embryo to form this foregut. So the foregut then is going to consist of the oral cavity to the pharynx level, really. And we know that the foregut really doesn't extend into the oral cavity because it terminates at the buccopharyngeal membrane. So it really is the oropharynx on down through to the larynx, where we know that the esophagus and the trachea join, and then we follow the esophagus to the stomach. We know there are curvatures and there's thin films of mesentery that stay attached to both its original dorsal and ventral sides, so these get flipped around and changed dramatically, and the term omentum is applied to them after all of these dramatic changes take place. Then, of course, after the stomach, we're really discussing the midgut, except for just a small portion, the first portion of the duodenum or duodenum. So some of the small intestine is included in the foregut, but the majority of the small intestine and large intestines are midgut, hindgut, and those will be in the subsequent lectures. We'll also spend some time discussing development of the accessory organs, more introductory today, and then in a little bit closer detail in future lectures. So the pancreas, liver, gallbladder, and spleen will be part of our discussion. So here we see an anterior view of an individual whose face is turned to the right-hand side, and we can identify the oropharynx just posterior to the tongue, so where the buccopharyngeal membrane would have been, opening down into the laryngopharynx, the larynx, the esophagus, and then the stomach, and then the first part of the duodenum will be the foregut. So we already see that the liver is probably going to be an accessory organ developing in relationship to the foregut. So this is our focus for today, and the remainder of the small intestine and the large intestine will be the focus of future lectures, even though we might refer to them today. So let's go to a higher magnification of the oral cavity and remind ourselves of some of the pertinent structures. We obviously recognize the tongue in the floor of the oral cavity, the mandible and the alveolar process with the teeth at the anterior portion of the oral cavity, but the margin of the oral cavity, of course, is at the lip. And this is the vestibule between the teeth and the gums and the mucosa lining the lip. We talked about the development of the hard and the soft palate and the formation of the tongue from a variety of tongue bud structures. And right about where the tip of the pointer is on this slide is the point where the buccopharyngeal membrane would have been attached, and it would come right up to the soft palate at the uvula in this area here. So this junction from the base of the tongue on up to the soft palate is where the buccopharyngeal membrane would have been located. So this portion of the pharynx just posterior to the tongue is the oral pharynx, and then just coming down below that towards the larynx and down to the main part of the larynx is the laryngopharynx. So from the tip of the epiglottis that we see here down to the first vocal fold, including the junction with the esophagus, is the laryngopharynx. So these are foregut structures. We already studied how the laryngotracheal bud extends from this foregut region early on and separates the respiratory system from the esophagus of the digestive system. We have always focused our attention as well on which germ cell layer contributes to the formation of what part of any organ, tissue, and cellular content of any organ. So this illustration of the adult, it's actually small intestine, but that's fine. It applies for, it's a generic one basically for all of the gastrointestinal system. And then as we proceed, we'll talk about some of the distinctions. For example, the stomach has an extra layer of smooth muscle. But let's start with the lumen first. We recall that the foregut was drawn up into the head and neck region and the future thoracic region by the head fold and lateral folds as well. And we'll see that in a moment. And the endoderm of the yolk sac becomes drawn up into that area as well. It doesn't get sucked up in there like we might be thinking. It just becomes encased by the structures moving down. We recall the pericardioperitoneal canal being that flat horseshoe-shaped structure, then the cardiogenic area, and the pericardial canal fold ventrally and then caudally and then dorsally up in to put the heart into the thorax. When that happens, it just encases the foregut. So the endoderm of the yolk sac is included within that. So we start with this portion then, thinking of endodermal origin. So there is an epithelial lining of the entire gastrointestinal system. It differs from one region to another, related to function, of course. So there is an epithelial layer and some underlying connective tissue. Connective tissue, mesodermal origin. We think of that immediately. And, of course, neurovascular bundle as well. And we know that there's lymphoid tissue in there as well. All of the mesodermal origin. So the surface epithelium plus the underlying loose connective tissue and a thin layer of smooth muscle also of mesodermal origin, is called the mucosa. So there will be a mucosal lining comprised of these three components in each case. Then there's some other loose connective tissue just deep to this innermost smooth muscle layer, the muscularis mucosa, and this is called the submucosa. So it is connective tissue with neurovascular bundle, therefore of mesodermal origin, except for the neural parts of all this. Remember, it's the nerve crest cells the neural crest cells that are generating the innervation of the gastrointestinal system. There are some nerve cell bodies here, the postganglionic parasympathetic neuron cell bodies and their dendrites are in here. You may recall the submucosal and the myenteric plexuses and ganglia of the autonomic nervous system, all formed by the neural crest cells. So when I say neurovascular bundle, remind yourself that the neural part is ectodermal origin from the neural crest cells. So keep that in mind, please. Except for the preganglionic parasympathetic axons in these plexuses are originating from the spinal cord, therefore from neural ectoderm. So I could torture you, but I won't. The neural always is the exception when I say mesoderm. That's the primary point. Then, of course, as we proceed farther and farther away from the lumen, we encounter more smooth muscle, and these are the prominent smooth muscle layers. There's an inner circular and an outer longitudinal smooth muscle layer, and where do they have that labeled? Here it is. No, that's the nerve plexus. Muscularis externa. So these two layers, the outer longitudinal and the inner circular smooth muscle layers, form the muscularis externa. Therefore, sometimes this muscularis mucosa is called the muscularis interna. And then the externa are the prominent smooth muscle layers. Then the outermost connective tissue layer is called serosa, especially when the mesothelial lining, simple squamous epithelium, is on the surface. There are some areas of the gastrointestinal system where that simple squamous layer of cells with a thin layer of connective tissue underneath, it's peritoneum, visceral peritoneum, will not be present, so there is not a mesothelium, and then it is called an adventitia, where the connective tissue in the outermost portion of an organ, say the aorta in the thorax is a prime example, its connective tissue on its outer layer, its adventitia, the collagenous fibers and elastic fibers and all that stuff, is just continuous with the surrounding connective tissue, loose connective tissue of the body wall. So we'll talk about some of the gut becoming retroperitoneal and losing some of its outer layer, the serosa, producing what's called effusion fascia. So we introduce it at this time so that you can see this serosal layer, thin layer of simple squamous epithelium that we're talking about. So we see the neurovascular bundle. Remember, lymphatics are included here, penetrating from the mesentery in deep to the serosa, or it'd be adventitia if the mesothelial layer was not present, and penetrating deep all the way to the lamina propria underlying the surface epithelium. If we back up for a moment to the surface epithelium, way back at the lumen, we see that there are some glands that extend from that, sometimes even into the lamina propria, and sometimes even into the submucosa, differing in different regions, and even different types of glands. Obviously, the glands of the stomach are going to be different than the glands of the small intestine, and so on and so forth. And then others will extend out of the gut tube itself, such as the duct of a gland outside the alimentary canal here. The pancreatic duct is a prime example here. So from this, we're beginning to imagine endodermal cells growing out. So an evagination out of its original site, either partially into the underlying structures, the lamina propria, or deeper to the submucosa, or even pushing through right out of the organ, the gut tube itself. So by starting with the adult appearance first, we can now almost begin to envision all the cells coming together, communicating, forming, allowing the neural, the establishment of the neural connections that were there very early on. We don't get all this developing and then finally the nervous system grows in. We've said all along that these connections are made very early when things are close together, and then you just elongate your axon to sustain your connection with your target site. So those structures are present from either the neural crest cell axonal extensions or migrating neural crest cells to form the ganglia, and of course the extending motor axon, visceral motor axon of the parasympathetic preganglionic fibers here as well. I understand if you don't know the autonomic nervous system really well, that's probably not making a lot of sense, but I continue to emphasize that distinction of the nervous system from the vascular system, including the lymphatics, developing from the local mesenchyme. And then the rest of the mesenchyme differentiating into all the other structures until we get to the epithelial layer and its associated glandular structures that was of an endodermal origin. So we have ectodermal, mesodermal, and endodermal contributions to the gut tube. Well, now let's back up a bit and think about the primordial gut after we've spent a significant amount of time on the adult gut tube. So it develops during week 4, so there's a lot happening in week 3 into week 4. We basically start week 3 with 3 layers, 3 weeks, the embryo, and in the 4th week we're already beginning to form the foregut. Remember that the oropharyngeal, I keep calling it the buccopharyngeal, but it's the same thing. Membrane is the anterior most or cranial boundary. It's safer to call it cranial because that tells us it's toward the head. And the cloacal membrane caudally. So it is an enclosed cavity up to the point that the buccopharyngeal, the oropharyngeal membrane, and the cloacal membrane begin to deteriorate. And remember, these two unique structures were sites where the original epiblast and hypoblast cells sustained their tight junctions with each other and didn't allow mesodermal infiltration in between them. So when we talk about the breakdown of these two membranes, it's just 2 cell layers thick. So that's not a challenge for the developing embryo at all. So the foregut then is the pharynx. We broke it down into specifically for digestive system, the oral pharynx and the laryngeal pharynx. Of course, there's the nasopharynx that really develops from the foregut as well. But I just left it out because we're focusing on the digestive system. The oral cavity is included, but it is, with exception to its development, it is not. The mucosa lining the oral cavity is formed from surface ectoderm on the branchial arches that are extending around and fusing. So I prefer actually just to leave the oral cavity out of the discussion of the foregut. But it's safe to leave it in there as long as we remind ourselves of its origin. Excuse me. And of course, it is inconsistent with saying that the cranial boundary is the oropharyngeal membrane, which is at the caudalmost portion of the oral cavity. So, that is an incongruity that should make sense versus the incongruities that I had on the exam this time. The glands, respiratory system are extensions of the foregut, as we had said. And I'm trying to stay focused just on the digestive system. Esophagus, stomach, proximal duodenum, liver, gallbladder, and pancreas. The midgut is the distal duodenum, the remaining part of the duodenum, all the rest of the small intestine, and the proximal part of the large intestine over to the distal 2/3 of the transverse colon. We'll see illustrations of this as we proceed, obviously. And the hindgut is from that distal 2/3, so we start then with the distal remaining 1/3 of the transverse colon and the descending and sigmoid colon. Also, some of the epithelium of the bladder and the ureters form from this hindgut area, but obviously are not part of the digestive system. Well, that brings us to this illustration where we remind ourselves that the cardiogenic region has now moved from that location of the pre- or prochordal plate way at the rostralmost end of the developing embryo. It is now moved ventrally, caudally, and then dorsally to become positioned within the primordial thoracic region. And in doing so, along with some lateral folding, it encases this portion of the yolk sac endoderm and creates the foregut. And the site of the liver development is going to be at the margin of that yolk sac, the yolk stalk, where the yolk sac will extend extraembryonically. We see that the body stalk is here, so at this stage, 3-week embryo, late 3-week embryo, the body stalk and the yolk sac haven't come together even with the lateral foldings to form the umbilical cord yet, so we're still just seeing the body stalk at this point. Thus, we focus our attention on these two margins of the attachment of the yolk sac, the yolk stalk, to the gut tube system that is forming, and these will be our margins to remind us of everything rostral to this rostralmost attachment of the yolk stalk. Yolk stalk is foregut, and it ends at the oropharyngeal membrane. And the stomadeum is the opening that goes into the future face and mouth region. So that reminds us that this is all surface ectoderm, and here it's going to line the oral cavity at the buccopharyngeal membrane or oropharyngeal membrane. We're going to have endoderm start there. So this is all foregut. So future pharynx, the very early laryngotracheal bud is just starting there, so the esophagus, and then somewhere in here the stomach is going to form, and the first part of the duodenum, and we know that the liver drains into the first part of the duodenum, so right at this attachment of the yolk stalk is where then we make a boundary. We go to the opposite side of the attachment, the more caudal attachment of the yolk stalk, and we can draw an imaginary line, and everything associated with this is midgut. And then from that imaginary line all the way to the cloacal membrane is the hindgut. So we have foregut, midgut, and hindgut all forming here. We see the neural tube has already begun to differentiate enough to recognize the brain vesicles up in the associated with the foregut region. Cranial nerves are going to innervate that, and then the spinal cord associated with the remaining part of midgut and hindgut, as well as foregut. And we know that the sympathetics, our preganglionics, are going to originate from sympathetic preganglionic motor neurons in the spinal cord. So that innervation, they've already extended their axons out to find their postganglionic neurons that developed from the neural crest cells and migrated around to become associated with the vasculature that isn't shown in this illustration. But we always started with the neural tube, and then we saw the dorsal aorta, so we know the dorsal aorta is present here, aorta, and any branches of it going to foregut, midgut, hindgut would be established quite early as well. So our attention today will be on foregut, as I'd said, and we'll mention the others just for purposes of reference. Now in the 4th week, of course, we're showing the more prominent dorsal aorta present and branches, the arch, the pharyngeal arch arteries that are present here in around the foregut would be associated with the neck region. And we can see the beginning of the trachea here, the laryngotracheal bud is developing. So that would be the level of the larynx, and the esophagus will branch from the larynx posteriorly, extending deep to the septum transversum, or caudally to the septum transversum. It is going to become part of the thoracoabdominal diaphragm. So we know just caudal to that location where we see this next branch of the dorsal aorta, the celiac artery, is going to be where the stomach is going to develop. After the stomach is the duodenum, and the liver will develop in this area and have a persistent duct system opening into the first part of the duodenum, and it drains into the first part of the duodenum. The development of the liver is more complex than just this endodermal contribution. This endodermal contribution is really going to form the biliary tree, and then there will be other cells that contribute to the stroma and parenchyma of the liver itself. So the celiac trunk, or the celiac artery, is the artery, the branch of the dorsal aorta that provides the arterial supply to this subdiaphragmatic foregut portion. And then just after where the liver bud is developing, we see the attachment of the yolk stalk, now pretty much encased within the umbilical cord, the body stalk and the yolk stalk, and a little remnant of the septum transversum being continuous with the extraembryonic mesoderm have encased all of these structures, and we're seeing the early umbilical cord. So that junction of the attachment of the yolk sac through the yolk stalk tells us foregut ends, midgut begins. We see the superior mesenteric artery providing the arterial supply to the midgut, extending out with the branch, the vitelline artery, going into the yolk sac. And then at the caudal attachment of the yolk stalk to the body structure, we know is the imaginary line for the termination of midgut and the beginning of the hindgut. And we see at the tail end of the hindgut we have the cloaca with a common opening into the digestive system and the urinary system that obviously separates. In humans, we have the separate structures, the rectum and the urinary bladder. But this is why we included the urinary system, parts of the urinary system, in the hindgut. The artery branching from the dorsal aorta to supply the hindgut is the inferior mesenteric artery. There are segmental branches that persist here in the future thoracic aorta, and these segmental branches of the aorta will give supply to the esophagus passing through the thoracic region. It's not until we get into the subdiaphragmatic region that we have these dedicated branches, celiac artery for the subdiaphragmatic portion of the foregut, the superior mesenteric artery for the midgut, and the inferior mesenteric artery for the hindgut. And obviously there's corresponding veins as well, and we won't get into the details of that right now. So this fourth week illustration, color illustration, very easily transitions right into this line drawing. And we see, obviously we can orient initially to number 14. That would be the oropharyngeal membrane. So number 1 is, of course, going to be the pharyngeal gut. The oropharynx is what we were referring to in our case, but it's really, at this time, undifferentiated between respiratory and digestive, so it's just the pharyngeal portion of the foregut. Number 2 is the tracheobronchial diverticulum. Number 3, obviously, is the esophagus. 4 is the stomach. We're beginning to see later in week 4 that it's already beginning to dilate. The posterior margin is growing faster than the anterior margin. We know that this is going to contribute to the lesser curvature on the anterior and greater curvature on the posterior. And then it flip-flops too, so we have to follow the development of the stomach to appreciate the adult arrangement. After the stomach, obviously, is the duodenum, and we see 2 outgrowths, endodermal evaginations, into the surrounding mesenchyme, and posteriorly is the pancreatic bud. Anteriorly will be the biliary bud, or the future gallbladder and biliary duct system. Obviously, 6 is the liver, developing in association with that biliary tree. After that portion of the foregut, the remaining duodenum, all the rest of the small intestine is midgut. We can see that number 8, the vitelline duct, persists. It is a connection of the yolk sac to the midgut. And then number 12 here would be the hindgut, obviously, and 10 would be the cloaca, 13 the cloacal membrane, as we saw. 9 is the allantois, or allantoic stalk that leads out to the allantois. And then that 8 was the vitelline duct. So a lot of structures to be identified and labeled on here. So plenty to keep us busy, just memorizing what's going on for the primitive gut in the 4-week-old embryo. So we saw that the esophagus develops from the partitioning of the laryngotracheal groove by the laryngoesophageal septum. That was discussed in the development of the respiratory system. So at that site of the laryngotracheal groove where that septum develops, we have a landmark to know from that point caudally to the subdiaphragmatic area is going to be the esophagus. By 7 weeks, it's going to reach the full and final length because the thoracoabdominal diaphragm becomes well established, and the stomach also will become dilated and take on its curvatures. The epithelium and the glands are derived from the endoderm, and then rapid epithelial proliferation actually closes the tube early on, and then ultimately there will be apoptotic cell death within the core of that solid column of cells, and there will be a reopening of the esophagus by week 9. The skeletal muscle is found on the proximal 2/3 of the esophagus, still of mesenchymal or mesodermal origin, but innervated by voluntary neural system. So if we get something kind of caught in our upper esophagus, and we can consciously bring it back up. Once it gets past about the halfway point, definitely down past that 2/3 point, you rely on the autonomic nervous system to bring it back up. So then it is involuntary to bring it back up. So the distal 1/3 is the smooth muscle, but both are originating from mesenchyme. And all the muscles are innervated by cranial nerve 10. This slide includes 2 images of histological preparations, cross sections of the human esophagus. And the first thing that cues you into this being the esophagus are the multiple layers of cells, each having their nucleus, very prominent, all the way to the surface cells. And this is, the surface cells are flattened. So this is a squamous epithelium. There's multiple layers, so it's a stratified squamous epithelium. It doesn't have dead cells on the surface, doesn't get calloused like the palm of our hands or the heel of our foot or any of the surface of our skin, obviously. So therefore it is non-keratinizing, stratified squamous epithelium, characteristic of the esophagus. There basically is only one other organ with the surface epithelium that is similar to this, and that is the vagina. But the epithelial layers are a bit more compacted. As we move from the lumen to the periphery of the esophagus, we can then follow this in the lower right-hand panel. And we can see the deepest layers of this surface epithelium have dark nuclei. We can almost make out what appear to be mitotic figures. So obviously the basal cells are very mitotically active in the adult to replace the surface cells that are lost. So as the new cells are produced from mitotic division of these basal cells and slowly make their way to the surface, they differentiate into the surface cells with all their tight junctions and things like that. And eventually they're going to die and slough off, and they have to be replaced continuously. Our gastrointestinal lining is replaced every 4 to 6 days. So these are very mitotically active in the adult. So all this epithelium that we've been discussing so far has an endodermal germ cell origin for the esophagus. The innervation is through the cranial nerve 10, the vagus. And then in this lower right-hand panel, we can see the underlying connective tissue. So there is lamina propria. Gosh, I couldn't remember that term. There's a lamina propria. There is not an inner smooth muscle layer, but there is the submucosa. So from the lamina propria up to the epithelium would be the mucosa. There is not an inner muscularis interna. Then there is the submucosa, and then here we see the smooth muscle layers. Wait, this is skeletal muscle. See how it's circular in appearance, and you have peripheral nucleus. So you can almost envision the circular cross-section of the skeletal muscle fiber and the peripherally placed nuclei. So this actually is skeletal muscle, so it's more so in the upper esophagus. It isn't the best example to show the inner circular and outer longitudinal layers, but they do exist. It becomes very well established, obviously, in the lower 2/3 where it's only smooth muscles. Next, then, we can discuss the stomach and its coverings. So it starts as a simple tube, but it dilates during the week 4 in the median plane, anterior and posterior surfaces, or ventral and dorsal. The dorsal border grows faster than the ventral one and creates the greater curvature, and the slower growing ventral border stays shorter, so it's the lesser curvature. So let's take a look at that. Again, we use our attachment of the yolk stalk as our landmark. Just cranial to that is where the stomach will be after we go a short distance for the first part of the duodenum, and the celiac trunk is providing the blood supply. So it's beginning to dilate here in this 28-day embryo. And when we take a cross-section, remove it in this type of illustration, we can see that the two body cavities, the peritoneal portion of the pericardio-peritoneal canal on each side has encased the endoderm and flattened onto its surface, leaving the dorsal mesentery all along its dorsal margin up to the dorsal aorta. And that dorsal mesentery obviously carries the neurovascular bundle. And then to the anterior body wall, or ventral body wall, is the ventral mesentery. So the stomach, the ventral margin is going to grow more slowly, and the dorsal margin is going to grow more rapidly. This is the 28-day embryo. By day 35, we see that accentuated greater curvature already, the original dorsal margin or border, and the ventral margin or border is not growing as rapidly and starting to form this concave indentation into the ventral surface. The ventral mesentery and the dorsal mesentery are still there. The dorsal mesentery is becoming elongated. The spleen is formed within it. That pancreatic bud has grown into between the two layers and has picked up its vasculature and is developing in the dorsal mesentery of the stomach, called the mesogastrium. So meso, mesentery, gastro, obviously stomach. So this is the dorsal mesentery of the stomach, also called the dorsal mesogastrium. and this would be the ventral mesentery or ventral mesogastrium. Here at 40 days, we can see that the dorsal mesentery, through this small restricted hole, is actually ballooning out to the left-hand side at the same time that the dorsal surface is moving to the left. So we have started with the stomach being anterior and posterior like this. Now the posterior surface is turning to the left and getting much longer, and as it turns to the left, the dorsal mesentery mesogastrium is going with it and forming kind of a pouch or a purse. It becomes an enclosed area with only a small restricted opening, and therefore it's called a bursa, an omental bursa, and this is the greater omental sac or omental bursa, attached to the greater curvature of the original dorsal surface of the stomach. And then the lesser curvature now is going to be the right margin of the stomach. And then it continues to contort as the small intestine elongates in the abdomen. It's a very ornate and highly orchestrated series of events, but eventually the distal stomach, the pylorus, becomes shifted to the right side, and the greater curvature, the fundus of the stomach, and becomes aimed toward the left side of the body. And here we see that dorsal mesogastrium folded and flopped upon itself, and what it will become is these two layers will fuse together as it continues to loop around, and it will become the greater omentum, a thin film of mesothelial membrane. So I'm going to stop on this slide because we're past our time. The remainder of this lecture is on the fall 2003 recording. There's only about 8 more slides. I appreciate your time and attention, and I hope you all had a good spring break, and I'll see you here on Wednesday.